In DNA replication, as cells divide, copies of DNA are produced that transfer genetic information into new cells. So, in DNA replication, the strands of the original or parent DNA molecule separate to allow the synthesis or making of a complementary DNA strand. The enzyme is called helicase, and this catalyzes or makes the reaction happen, right? And so this makes the unwinding of the double helix by breaking those hydrogen bonds that we talked about between those complementary bases, A, T, C, G. And the resulting single strand acts as a template for the synthesis or creation of a new complementary strand of DNA. In DNA replication, the separate strands of the parent DNA are templates for the synthesis of two complementary or matching strands, and this produces two exact copies of DNA. And sometimes these are called daughter strands. In the complementary base pairs, as they come together, the DNA polymerase catalyzes the formation of a phosphodiester bond between the nucleotides. Basically, it glues it together, okay? And then the entire double helix of the parent DNA is copied. The DNA polymerase is going to recognize the area that's going to be complementary and start making it. Okay, so anyway, but it's going to be copied. And then this process produces two new DNA strands, and these are identical to each other and have the exact copies of the parent DNA. And these, again, are going to be called daughter DNA. So complementary base pairing ensures that the correct placement of DNA of bases are going to be in the new DNA strands. So how many hydrogen bonds are formed between each of the following base pairs? Okay, remember back to either the 2 hydrogen bonds or the 3 hydrogen bonds from the previous video. So pause me and come back. All right, the A to T, these are going to have 2 hydrogen bonds that are going to be broken, where the G to C, those are going to be 3 hydrogen bonds that have to be broken. All right, so why do we care? Well, this is DNA fingerprinting, okay? And so in a process that's called DNA fingerprinting, the enzymes are used to cut DNA strands into smaller sections. And the resulting fragments are separated by size, and they're treated with a radioactive isotope that adheres to specific base sequences. And the resulting pattern of bands is known as a DNA fingerprint. So one application of DNA fingerprinting is going to be in forensic science. Okay, and this is where DNA from samples such as blood, hair, or semen is going to be used to connect a suspect with a crime. So we gotcha, right? So in dark and light bands on X-ray films are going to show up as a result of the radioactive isotope that's going to adhere to specific sequences. Okay, and so we are going to label the suspect, right? And then we're going to be looking for the different bands to see which ones are identical and then be able to catch bad guys. Right, awesome.